The genomic library in the gene source, one of the most important thing is the genomic library. There are the various sources for the gene, and among that various sources of the gene, the genomic library is one of the most important source. Now, what is this genomic library? Many times we heard that name library where there is a collection of number of books. But in case of this library, there is a collection of number of genes, and that collection of number of genes or our gene of interest, that is nothing but the genomic library. Now in this genomic library, what is this genomic library? Now this genomic library is nothing but the mixture of clone which when derived directly from the genomic DNA called as the genomic DNA library. Now this genomic DNA library that must contain at least one copy of every DNA sequence in the genome. Means whenever we construct the genomic library, there should be the presence of at least one copy of the every DNA sequence, as during the formation of or during the construction of genomic library, there is a formation of number of DNA fragments, and in that entire genomic or genetic material, whenever there is a formation of DNA fragments in that genomic library, there should be at least one copy of the every DNA sequence in the genome. Now this genomic library is prepared by using the restriction endonuclease enzyme. This restriction endonuclease enzyme plays very important role in construction of the genomic library, because this restriction endonuclease enzyme which cut the DNA at the specific site, as this restriction endonuclease is one of the most important tool which is used in the recombinant DNA technology. Now in this genomic library, cloning of the entire genome in the form of the library of a random genomic clones, that is called as the shotgun experiment. Now this, according to the shotgun experiment, there is the cloning of entire genome in the form of the library of a random genomic clones, and this experiment is fragment of varying sizes having the cut of different places of the genome that lead to cut at the inconvenient places. So the fragment having complete gene will difficult to obtain. To overcome this problem, there is a strategy that is used in the shotgun experiment, and that strategy is the use of restriction endonuclease enzyme which have a short recognition sequence. Means if there is a use of the short recognition sequence, means the recognition sequence with a few nitrogen base, then this problem or this strategy can overcome that particular thing. Now how there is a construction of the genomic library? First of all, there is a total genomic DNA is extracted. Now as we know that any organism which have a genetic material, and that entire genetic material is called as the genome, and this genomic DNA is extracted. First of all, there is the extraction of the genomic DNA. This extracted DNA then broken into the number of fragments, and these number of fragments are formed by using the restriction endonuclease enzyme. Now this restriction endonuclease enzyme having a 4 base or the 6 base recognition sequence. This 4 base or 6 base recognition sequence for the restriction endonuclease enzyme that plays the very important role in the construction of the genomic library. If we consider that the 4 base sequence is occur, means if there is a 4 nitrogen base recognition sequence, this means that is 4:4. So this recognition sequence which occur in that genome, that is after the 256 base pair, and if there is a recognition sequence of the nitrogen base, that is 6 nitrogen base, that is 6⁻⁶, that is 6⁻⁶, that is 4096, means this is occurred after this 4096 nitrogen base. Now here these two things, among that two things, that is 4 and 6, fragment produced by a partial digestion with an enzyme of 4 base recognition sequence are the appropriate for the cloning of that particular genetic material. Means these 4 base recognition sequence, restriction endonuclease enzyme recognition sequence are useful for the cloning purpose during the construction of the genomic library. Now there is a single or mixed digestion. Single digestion or the mixed digestion, there may be the use of a single restriction endonuclease enzyme to make a number of DNA fragment, or there may be the use of the mixture of that restriction endonuclease enzyme. For example, ALU1, HAE3, there may be the mixture of different restriction endonuclease enzyme. Now this partial digestion, that also plays very important role during the construction of genomic library. Partially digested DNA that subjected to the DNA electrophoresis, means whatever the number of DNA fragments which are formed with the help of the restriction endonuclease enzyme by the partial digestion, these DNA fragment that must be separated before the transfer into the vector and then vector into the host cell. So the first important thing, there is these partially digested DNA subjected to the DNA electrophoresis. What is this electrophoresis? Electrophoresis is nothing but one of the most important process where there is a separation of the charged particle in response to the electric field. And here, as we know that DNA has a particular charge, that is the negative charge. This shows the migration, means the number of DNA fragments which are formed, that is separated with the help of DNA electrophoresis. Those DNA fragments which are separated with the help of DNA electrophoresis, these DNA fragments then inserted into the vector. Means whatever the DNA fragments which are separated with the help of this DNA electrophoresis, that is inserted into that particular vector. Now the vector which are used for specially for the construction of the genomic library, these vectors are the lambda or the cosmid vector, that is used for the cloning, because this lambda or the cosmid vector, this has the more capacity to insert the gene of interest. Near about 25 to 30 kb size of the DNA fragment can be inserted into that particular vector. That's why this particular vector may be used for the construction of the genomic DNA library. Now this vector containing the DNA insert that clone into the bacterial host, means that whatever the DNA insert which is insert into the vector, that is clone into the bacterial host for making a number of copies of that particular gene of interest or the DNA insert. Now here in this picture we can see how there is the construction of the genomic DNA library. This is the cell from which there is the extraction of the DNA. That extracted DNA that is digested with the help of restriction enzyme, that is restriction endonuclease enzyme. That restriction endonuclease enzyme forms the number of DNA fragments, and this number of DNA fragment along with the use of this vector, that DNA fragment inserted into the vector. Now this is the recombinant vector, and this recombinant vector then transform into the bacterial cell, and this bacterial cell that grown into the media. So due to which there is a formation of the number of copies of that particular DNA fragments. So in this way, where there is a collection of the number of DNA copies or number of that particular gene of interest or that particular DNA fragments. Because of this collection of the number of DNA fragment,